Hello, this is Dr. Pickett. This is the audio narrative for the last part of my Lecture 3, starting at slide 63, and then progressing into Lecture 4. I might post these as two clips, we'll have to see at this point. This lecture will substitute for the lecture on Thursday, January 31st, that we would have had if we weren't plunged into the polar vortex. God bless us. Okay, so this is starting with the slide titled X-linkage in humans, Haemophilia impacts X hemizygotes. So before we discuss the inheritance of X-linked traits, we need to talk about this phenomena of hemizygosity in comparison to heterozygosity or homozygosity. Because of the sex chromosomes, humans have the possibility, obviously, of two chromosomal sexes, the XY males and the XX females. Again, that's the species-typical situation. There are lots of variants that we'll discuss later on when we talk about chromosomal variation in terms of the karyotype. So females, because they have two copies of the X chromosome, have the possibility of being heterozygous and homozygous, either heterozygous dominant and trans to a recessive, or homozygous dominant or homozygous recessive. Males have a unique situation, however, though, because much of the X chromosome is composed of genes for which there can only be alleles on the X because the genes simply do not exist on the Y chromosome. There are parts of the Y chromosome that do have similarity to the X's. That allows these two chromosomes to pair during meiosis and undergo normal meiosis 1 segregation. But very rarely do you see evidence of recombination. If you do see evidence of recombination between the X and the Y, this often creates what are referred to as variant chromosomes, where a piece of the Y becomes attached to the X or another chromosome. So, thus, males have the possibility for the X chromosomes of being hemizygous, one copy of a gene on the X, no copies of the gene on the Y, which contrasts with females who can be homozygous and heterozygous for genes that are found on the X chromosome. So in that situation, we have a very unique pattern of inheritance of traits in families with X-linked genes. This example is the X-linked hemophilia that is present in the royal families in Europe, in part because, as we discussed last time, of the phenomena of consanguinity, where individuals that have some level of relationship do mate with each other. This is very common in royal lineages, presumably because royal families were trying to limit the diffusion, if you will, of wealth, privilege, power outside of the family, and we're trying to concentrate wealth and power within the family. And so you had these families in Europe that had broad patterns of connection. Many of them can trace their family lineage back to a shared common ancestor, Queen Victoria, who is individual Roman numeral 2 dot Arabic numeral 1 in this pedigree. Queen Victoria came from a lineage that was fairly well documented. There is no history prior to her of hemophilia, which is a clotting disease, a failure in individuals that have wounds to do normal blood clotting. Also because there is a failure in blood clotting if you have tears within the body, if you have organ damage, etc. Males that have this trait have a tendency to have catastrophic both internal bleeding and bleeding outside the body if there is a wound that impacts the surface of the body. So anyway, because so many distinct members of the royal family could trace their lineage back to Queen Victoria, and apparently with Queen Victoria there arose within her oogonial stem cells or stem cell populations a mutation on one of her two X chromosomes that causes this clotting disorder, hemophilia. As Queen Victoria entered into menarche, began to have children, and produced a large number of children. If we see in this pedigree, she had individual 3.1, 3.3, 3.4, 3.6, 7, 8, 9, 3.10, and 3.11. So she was the mother of 11 children. Of those 11 children, it is evident that there are at least two daughters, individual 3.4 and 3.12, who were capable of transmitting hemophilia risk-associated alleles on their X chromosome, on one of their X chromosomes, to their offspring. How do we know? If we look at, for instance, the offspring of a mating between individual 3.4 and a male from outside of the family, individual 3.5, we see that this mother gave rise to a son who had hemophilia, and also two daughters. One daughter, individual generation 4, individual .6, is a scored carrier, an evident carrier of hemophilia, because she gave rise in generation 5 to two sons who were affected. And then one of these individuals, individual 4.11, is Tsarina Alexandria of Russia. Tsarina Alexandria of Russia, marrying Tsar Nicholas, gave rise to Prince Alexei, individual 5.10, who was also hemophiliac and who was, shall we say, through his disease, the infamous conduit of the mystic Rasputin into having an influence in the royal family of Imperial Russia. So this is fascinating. What are we seeing with this X-linked trait? Well, we're seeing Queen Victoria giving rise to individual 3.10, a son who was affected with hemophilia. This son was able to transmit hemophiliac risk to individual 4.15, a female unaffected but an evident carrier. Why? Because she is scored by her genotype is scored because of the fact that one of her male offspring developed X-linked hemophilia. What we see is a pattern in which males present as affecteds, whereas female carriers are inevitably the mothers of those affected males. Why? Well, remember, males can only transmit their Y to their sons. Thus, if a couple has a son, and that couple has X-linked or X-associated hemophilia, it is inevitable that his mother provided the disease-associated allele on the X that entered into her egg during meiosis and was then fertilized by the father's sperm. So we see a fascinating situation. We see a phenomena of frequent generation skipping. An affected male gives rise to an unaffected but carrier female, who gives rise exclusively to affected males. And this is perpetually the case. Mothers who are unaffected give rise to affected males. Thus, we see routine generation skipping. Now, I have provided a fairly simple animation that just shows, for instance, if we move to the next slide, slide 64, Queen Victoria as a heterozygous carrier. It is almost certainly the case that her germ cells, at least some of her germ cells, were heterozygous. We don't know if she might have been a genetic mosaic. There's some evidence that suggests that might be the case. But at this point, we can assume that the germ cells that we care about were heterozygous. as those germ cells underwent meiosis, we see, for instance, one of her eggs was fertilized by a sperm that gave rise to male 3.10. That male, as a hemizygote with the hemophilia associated mutation on the X chromosome and then the Y chromosome, is affected because the only blood clotting gene that this individual has is a mutation that is broken, and thus they don't have normal control of bleeding. However, she also produced two heterozygous females, individual 3.4 and individual 3.12, neither of which are affected by the disease, but both of which transmit it. They see it first in their male offspring and in the production of carrier females. If we continue to move down, moving to the next slide, we can see, for instance, in the cross between 3.4 and 3.5, a heterozygous female mates with a homozygous unaffected male. For male offspring, the male loads the Y into his sperm when the egg is fertilized. If an egg contains mom's unaffected X chromosome, for instance, male 4.9, that is simply a male that is hemizygous, wild type, or with a normal allele. However, by happenstance, in a case of male 4.10, the mother's affected X chromosome is loaded into her egg, the father loads the Y into the egg, and thus this male is a hemizygote if we move to the next slide, and they present as affected. They have one copy of the hemophilia locus on their X, no complementary copy on the Y, and so because of that, they are inevitably affected by disease. So with X-linked linkage, some of the highlights are unaffected but known carrier mothers having a 50% chance of producing an affected male offspring. Affected males, alternatively, have no probability of transmitting their disease to their male offspring because they're loading their Y into the sperm that gives rise to all male offspring, not the X. But males who are affected do have a 50% chance of producing female offspring that are at least heterozygous for the disease-associated allele. Now, why don't we see affected females? Well, remember, affected females would require a mating between a carrier female and a hemizygous affected male. Thus, they tend to be very, very rare in society. There are clinical reports in the literature of women with X-associated hemophilia, and thus, if you have a female that is hemophiliac, do not discount the possibility that they might have an X-associated disease, an X-linked disease. But they're very, very rare because they require this unique situation where a heterozygous carrier mother or a homozygous affected mother, although I know of no instances where that has happened, where you have a heterozygous carrier mother mating with a hemizygous affected father. So with the vast majority of X-linked disease traits, we see affected males in the family, generation skipping, and it is obligate that the disease-associated allele was inherited from the mother. It is impossible that the allele was inherited from the father. So I just want to go to the last slide in this deck. We've talked a lot now about the Mendelian case, the situation in genetics that we see from Mendel's early paper, now from his notes as they have become slowly available to the scientific community. Mendel did not discover some of the phenomena that we're going to talk about in just a bit. He did discover a very interesting phenomena, uniparental inheritance, but unfortunately he did not have the tools at the time. He didn't have sophisticated histology. He obviously did not have access to molecular biology and biochemistry. And so, although in the experiments that he did, unfortunately, with the plant buckthorn, he made an amazing discovery that it is possible, for instance, for there to be uniparental inheritance. He simply did not have the tools to interpret it, and he began to doubt the validity of his own work because these follow-on experiments seemed to disprove the now law or proposition of segregation and the law or proposition of independent assortment. It's interesting, he turned in his later life to doing more administrative work in the Abbey. He did do teaching. He also did mentoring of young priests, but he tended, as far as we understand, to turn away from original investigations in biology. So now that we have the Mendelian system under our belt, we understand segregation, we understand independent assortment. We see in the experiments that we've discussed alleles that are dominant over alleles that are recessive. That is, in a heterozygote, if there is a dominant allele, the dominant allele presents in the phenotype. The recessive allele is still present in the genotype, but will not reveal a phenotypic impact unless there are further generations in which it is possible to produce a homozygote. So anyway, I hope that's somewhat helpful and gives us a little bit of a summary of our, shall we say, quick march here in the first few weeks of class through classical Mendelian genetics. But now we're entering into Lecture 4, where we're going to discuss situations in which our expectations from what we now know from this point are going to be violated. I call these violations and exceptions of the law of segregation, independent assortment, simple dominance, and recessiveness. Now we're going to see that some of these violations lead to ideas like the fact that each chromosome has many genes on the chromosome, not single genes. Single genes were behaving as hereditary characters in Mendel's experiments. Now we're going to see that these genes are queued up, are lined up in a long physically connected chain on the chromosome, and thus chromosomes can have many genes, many alleles that are affecting diverse processes in biology. This will give rise eventually to our understanding of genetic maps and even of how we generate pseudomolecules in the Human Genome Project and why we generate pseudomolecules in the Human Genome Project. We're also going to come up with a new concept of alleles. Why will that be important? Well, up to this point, we've kind of talked about alleles in sort of two flavors, dominant or recessive. You may have also heard me describe these as wild type and mutant, potentially. Eventually, we're going to begin exploring alleles that are not fully working or fully broken, but they're partially broken, or they have other types of roles. They're dominant because they're doing something novel in the physiology of the system that's under consideration, of the individual bodies that we're talking about, be they flies, peas, or man. So we're now going to kind of move away from our comfort zone, simple dominance and recessiveness, simple segregation, independent assortment, and we'll begin to look at other situations which we kind of broadly classify as non-Mendelian behaviors. These are situations where allelic interactions don't show simple dominance. We may not see independent assortment. Luckily, because we have a firm grounding in Mendel's tools, we'll be able to come up with explanations. We'll be able to come up with classic statistical models that we can test with frequentist data that we'll generate, and we'll see that even though maybe Mendel's version 1.0 ideas are not applying, the deep understanding that we have, alleles of a single gene segregate on their homologs in meiosis 1, alleles of different genes assorting independently, all of that biology will still be here. So the nice thing about that is that will give us lots of wonderful statistical predictions that we can make about the frequencies with which different phenotypes and genotypes should appear. But Mendel, I'm afraid, would have been quite baffled by some of the phenomena that we're going to see. Hello, my friends. At this point, I'd like to begin my slide deck lecture 4. We'll see how far we get through this. I'm going to try and do this as a normal 75-minute lecture, and we will assume that at that point you'll probably be tired of listening to it. All right. So, before we kind of begin talking about some of the new exotic allele interactions that we're going to discuss in the context of kind of non-Mendelian types of genetics, I think one of the things we need to do now is sort of expand our idea of what we mean by alleles. We've been in kind of a binary, a simple two alternative universe thinking about alleles up to this point. In that universe, what do we mean? Big A, little a, what does that mean? Big A may be the species typical wild type allele, what we encounter in a population of Drosophilas, peas, or humans in the wild, as it were. The allele that is perhaps at the highest frequency in one of those populations. In a situation where we're talking about a biomedical, a gene with biomedical implications, we may say this is the allele that is not associated with disease. It is the allele that may be protective against disease. Thus, in a way, we kind of think of the wild type allele as our standard gene. It's functioning normally. The RNA and protein that are produced using the encoded information in that gene plays a normal role in the physiology of the organism. Now that contrasts at some level to little a. Little a, often the recessive allele, is an allele that we may encounter rarely in the broader population of whatever organismal species we're dealing with. It is the allele that may be the disease-associated allele often in humans, although we'll see that that's not universal. We'll often describe that allele biochemically as not producing a normal mRNA and not producing a normal protein, i.e. The physiology, the cellular and organismal physiology of an organism that is homozygous recessive is missing an important piece of its biology because the information at that gene is corrupted, and thus the organism that is homozygous recessive has a missing component in their normal biological function. So there we kind of have two alleles. How do we describe these? The wild type allele, species typical normal function. The mutant allele is often an allele that from a functional perspective we might describe as a complete loss of function, or using the language introduced by Herman Molar and his system for allele description called allelomorphy, we might describe that allele as an amorph. Amorphic alleles are complete loss of function alleles. In the modern world of biochemistry, what does that mean? That gene is fully broken. We're not getting normal protein decoded from the information at that gene. We're not getting potentially a normal RNA made. So let's go through this system of Mullerian allelomorphy in detail. First, I'd like to introduce Herman Muller to you. Herman was a bit of a, shall we say, local cause celeb at Indiana University in Bloomington, where we had seminars discussing some of Muller's ideas. We had seminars discussing mutation. We had seminars discussing, in fact, I saw one of my first seminars discussing the impact of high energy radiation on genes under the guise of one of the Mullerian seminars that we discussed. So Herman Muller was an interesting character, and I think one of the things he acts as is a bit of an inspiration for young people who might think they need to be on, shall we say, the simple, straightforward path in life in order to ultimately succeed. I think one of the things that Dr. Muller shows is that you can have a variety of lives. You can have a variety of things that you try which may or may not succeed. You can explore ideas that may or may not be ultimately good ideas, but it's okay to be wrong. It's okay to question yourself, to more deeply query and challenge your own hypotheses and opinions, because in a way, ultimately, that's the way you know you're getting closer and closer to a true description of nature. If we never doubted our work, if we never tested our hypotheses, if we never went through a period of time where we were uncertain, it is unlikely that we would ever get to the best explanations for the natural phenomena that we see. Now, Muller was also a very colorful character as he was exploring these diverse multiple lives. He was a veteran anti-fascist. He even went over to Spain during the Spanish Civil War and contributed both kind of at field hospital levels, but also by doing research in various types of traumas that required transfusion, trying to support the anti-fascist movement in Spain. He interacted with some of the individuals in the International Brigade, the Lincoln Brigade from the United States, who again were transitioning to Spain. One of the things that he became interested in Spain was the political philosophies that we would now associate with kind of classical state socialism. And in fact, he flirted with communism and became kind of a dedicated communist as he was working more and more with the socialist groups that were fighting fascism in Spain. An element of that is that he decided to continue his work in the Soviet Union. Thus, he emigrated to the Soviet Union. He opened a lab in the Soviet Union where he began to do Drosophila work and other types of genetic research, hoping to contribute to building a solid scientific foundation for genetics, for Soviet genetics. Unfortunately, Herman Muller came right up against a young fellow who was very attractive to the political leadership of the Soviet Union, Trofim Denisovich Lysenko, who espoused the ideas of the inheritance of acquired characteristics. Now, I know now we talk a bit about modifications of gene expression, modifications of gene regulation that can be potentially at least inherited through the mitotic lineage from cell to cell, and may in some instances have some inheritance across generations. We describe this as epigenetic modifications of DNA. But Lysenko assumed that the genes themselves were friable, that a gene could somehow sense an environmental change its nature, change its allelism, and then that allelic change would be transmitted from generation to generation. We know that this does not happen. Epigenetic modification is fascinating, but for Lysenko's ideas, and even being charitable, interpreting some of Lysenko's ideas as epigenetic, at the level that he was doing analysis, it was simply pseudoscience. There was no evidence for any of the scientific ideas that Lysenko had. But unfortunately, Lysenko went to war with what he described as the Mendelists. Literally, Soviet geneticists' labs were burned. Their scientific materials were destroyed. Their Drosophila, their plant, even some of their animal strains were destroyed. So Lysenko championing as a political idea some of the valuable things for state agriculture that epigenetics might get, decided that genetics had to not be true, took a political stance that it was not true, and literally drove genetics from the Soviet Union, setting Soviet biology back perhaps almost 50 years, almost a half century, through this political pseudoscientific idea. Well, encountering this unfortunate situation led Muller and his family to quite literally flee the Soviet Union and then immigrate back to the United States, bouncing around University of Texas, other universities in the United States. He began to encounter some resistance because of the then emerging anti-Soviet, anti-communist feeling in the United States. Muller was blacklisted as a communist. That means that because of his past political affiliations, various types of scientific and other opportunities were withheld from him. But luckily, he was given a chance by a very visionary university president at Indiana University Bloomington, Herman B. Wells, who really valued scientific truth, valued scientific observation, gave Muller a safe place to do his Drosophila work. And through that, Muller became one of the founding members of the genetics group at Indiana, and because of the support of Indiana University, identified, for instance, high-energy radiation as mutagenic, as genotoxic. These discoveries with X-rays and other types of radiation and their impact on the genetic material eventually led Muller to being awarded the Nobel Prize in 1946, one of the 4 Nobel laureates in biology that can trace either their training or their professional work to Indiana University biology. And I note that although many of our famous Chicago universities who will remain unnamed, none of them at this point can hold a candle to the Nobel history of that doughty small university in central Indiana. So I enjoy kind of talking about Muller, a colorful individual, many setbacks, many interesting ideas, some good, some bad. He had later ideas about human genetics that I think were, although current at the time, we would now see as being quite deeply flawed from an ethical perspective. But you can be somebody that has, shall we say, a colorful history, and you can still get that PhD, you can still get that Nobel Prize. You don't have to be on the simple, narrow path to get to your destination. So I think he can be a bit of an inspiration for young people who may sometimes feel very constrained in their own lives to explore other possibilities, other careers, take chances and risks because they may be afraid that opportunities only come around once. Certainly that can be a risk, but Herman Molar stands as a counterexample that you can take risks, you can have setbacks, you can have people autoclaving your flies and burning your lab, and you can still end up sticking the landing, shall we say. So Molar's, outside of his Nobel work, one of his key insights was the idea that alleles don't describe a simple binary system, that in fact there are all different types of functional classification of alleles that you can encounter in the wild. Now, we now describe these alternative forms of genes as alleles. If we now move to the next slide. So this is slide number, this is slide number 4 in the deck, labeled section 4.1. These alternative forms of the genes are quite fascinating. We call them alleles. This simply means that the genetic information that they encode, although they are at the same spot on a chromosome, they are the same gene, the encoded information is different from allele to allele to allele. The sequence of DNA may be different. The activity of the gene may be different. What are the sources ultimately of alleles? Mutation. Changes in the DNA sequence that propagate into changes in mRNA accumulation or the sequence of mRNA that propagate ultimately into whether or not a functional protein accumulates with an expected amino acid sequence or maybe a corrupted protein accumulates, etc. So mutations are changes at the DNA level whose effect are ultimately seen kind of propagating up from the genotype to the phenotype, because when a gene is corrupt, an mRNA is corrupt, and then a protein is corrupt. I've described the wild type allele as the one that occurs most frequently in a population. Often these are dominant in relation to the broken recessive alleles, but that isn't always the case. Thus, these are usually but not always dominant. So what do we mean again by mutation? We mean that the genetic information is modified. If we transition to the next slide, often that produces an altered gene product, but by that we mean altered in an almost infinite number of senses. There may be no gene product produced at all. No protein accumulates. The gene is so fully broken, it can't do any mRNA accumulation or any protein accumulation. It may be corrupted at a level where you have just a few changes in the DNA bases that causes a few changes in the mRNA and perhaps a few changes in the amino acid. This broad difference in terms of effect may be a continuum where you have a wild type allele versus a fully broken allele on the two poles of the continuum. We now know that there are alleles that lie along that continuum that have many different variations of change or corruption in relation to the wild type allele. So new phenotypes might result because of these genetic changes. We might eliminate an enzyme function, an enzyme required for normal life and development. A mutation may cause that protein to not accumulate and thus that function to not be offered. If you've taken cell biology, you know how cell biological pathways are proteins touching proteins, touching proteins, touching proteins before finally a job in a cell is done. Well, if one of the proteins in that chain of interactions is missing, the entire pathway may not happen, and you may not have secretion of an important hormone from the cell or a receptor on the cell surface being able to signal to the internal cytoplasm of the cell in order to do its job. So mutations can have dramatic effects all the way from the level of cellular processes to the level of functions of organs to the level of appearance, activity, viability, and flourishing of individual organisms. So how do we describe these up to this point? We've kind of used a very simple nomenclature where dominant alleles are designated by capital letters. Recessive alleles are displayed by lowercase letters. That's something that you'll see fairly commonly. If we move to the next slide, geneticists do use a variety of symbols for alleles. Here we're in section 4.2. Dominant alleles, usually indicated by either an italic uppercase letter or an italic letter or group of letters. Some may be upper and lowercase, but you'll have a superscript of a plus symbol, a positive symbol, a plus symbol from mathematics. Recessive alleles are usually indicated by either an italic lowercase letter or collection of letters. You may also have lowercase letters with a superscript minus. These change from organism to organism, literature to literature. Sometimes you will have a situation where by shorthand, wild-type alleles for all of the alleles in consideration will just be shown with a single plus. So the mutant allele might be little d. You might see a chromosomal description, little d, a line, a horizontal line, then under the horizontal line plus. What does that mean? That's a big D, little d heterozygote. It's confusing because the big D plus has been shrunk down to just the plus symbol, but because little d is shown, we know that we have a mutant recessive allele of D designated by little d. We know we have a wild type allele designated by plus, but by shorthand, people are just not showing you the big D. In situations, if we move to the next slide, section 4.5, where there's no dominance interaction, and we'll see this in just a bit, there can be allelic series in which alleles do not show simple dominance interactions. The human blood group, which we'll discuss towards the end of the lecture or towards the start of the next one, is a fascinating situation in which alternative alleles both fully show themselves in the phenotype. So in that case, there is no dominance. We call it codominance. You can be big A prime, or big A other symbol. The phenotype is big A prime, because one of those alleles is not dominant to the other. Both are shown. Often, if no dominance exists, italic uppercase letters and superscripts are used to denote alternative alleles. But if we said something like here, you'll see there's R1 and R2, two possible alleles at the R locus. The possible genotypes are R1, R1, homozygous R1, R1, R2, heterozygous R1, R2, or homozygous R2. The heterozygote, the phenotype is R1, R2. The phenotype is not R1 or R2. Both alleles fully manifest themselves in the phenotype. So, now that we've gotten a little bit of a discussion about allele designations out of the way, let's kind of pull back a little bit more and talk about now with the next slide Mullerian allelomorphy, how we describe from a functional perspective the different coding possibilities, functional possibilities of alleles that we may encounter. Well, these sort of function schemes are interesting. With allelomorphy, almost everything will end in the concept morph. It'll end in the word morph. You'll have hypomorphs, amorphs, etc. They're talking about alleles with different functional potentials. In more modern literatures, you'll see folks switching to what's called the function system of allele designation. Does a gene have a normal function or wild type function? Is a gene a partial loss of function, i.e. in relation to the wild type allele? the gene product of a partial loss of function allele does the normal job of the gene less well in the organism, or is it a complete loss of function allele? Depending on the literature you read, you may see morphe descriptions for alleles or function descriptions for alleles, but they are broadly similar and in most cases are interchangeable. In your book, there is a tendency to use both types of allele descriptions, so I do try in this lecture to draw equivalences where I can. So let's start out with kind of the classic recessive allele, an allele in which the gene it is describing, the allele that it is describing, is completely broken. In relation to the wild type allele, there's absolutely no possible biochemical or molecular activity. We describe these types of alleles as amorphs. They produce no or very little normal protein. These are usually recessive. Using function speech, they may be called complete loss of function alleles. They are also often referred to in some literatures as null alleles. Amorph equals complete loss of function equals null. Depending on the literature that you're viewing, those are synonymous. Now, what about alleles that have certainly undergone some damage, but they're still able to kind of do the normal job that the gene can do at least some reduced level? You know, we might think about this as like a like a block stacker protein. If you're thinking about a perfect Lego stacker protein, that enzyme, Lego stackase, can put together 60 Legos by snapping them together in a second. It's an incredibly good Lego builder. It's an adult fan of Lego. So it can assemble huge Lego structures very quickly. Well, a hypomorphic Lego stackase allele might only be able to stack 2 bricks per second. Well, if we think about stacking bricks, in 1 second, Lego stackase wild-type allele is going to connect 60 Legos. The hypomorphic or partial loss of function, non-null, non-amorphic, but partial loss of function alleles is going to take 30 seconds to make the same stack of 60 Legos. So in this situation, you get some protein made, some RNA is made, but there's a little corruption of the information, so although the protein has the potential to do some aspect of the normal job of the gene, it simply is not as efficient or as capable by some measurement. So hypomorphic alleles, partial loss of function, those are often used interchangeably in the literature. Now, let's talk about dominant alleles. Often, the null or amorph and the hypomorph or partial loss of function alleles will behave as recessive alleles in comparison to the wild type allele. So the wild type allele will be dominant to either amorphic or hypomorphic alleles. Hypermorphic alleles are interesting. If we think about the Lego stackase enzyme and its 60 connected Legos per second, a hypermorphic allele does the job of the wild type gene too well. From the perspective of Lego stackase, maybe it can stack 180 Legos per second. Rather than the wild type gene doing its steady stacking, this is the frenetic, anxious, hyper gene that can just stack like crazy. Now, you might think that doing the wild type job better than wild type would give this allele a positive impact on the organism, but often we find these alleles are too much of a good thing. The cell fills with Legos, and so those Lego stacks and their components excuse me, are not available for other jobs in the organism. Given that situation, the hypermorphic allele can also have a negative impact on the physiology, viability, vitality of an organism. Even though it's doing the wild type job better than wild type ever did, it may still be negative for the cell. Hypermorphic alleles are often dominant to the wild type allele. If we have them on two homologs in a heterozygote, the phenotype will be associated with the hypermorphic allele. The wild type allele will be recessive, and so the phenotype of the wild type allele will not be contributing to the phenotype. Using function speak rather than morphe speak, we often refer to these alleles as gain of function alleles. They're doing a new function. They're doing more of the normal function. They've gained activity in relation to the wild type allele. Now, we have an interesting class of alleles called the antimorph. Antimorphic alleles are interesting because they usually function in situations where you have dimers or multimers, many copies of a protein, the same protein coming together to make a molecular machine. Here, the wild type allele, the proteins made from the wild type allele will assemble into a normal machine, which will then go and do a job. The antimorphic allele will produce a form of the protein, but if it comes together with the wild type copy of the protein, it kills the multimer. It actually functions as a molecular poison, making it so the wild type protein can't be with a partner able to do its job. The antimorphic protein acts as a bad partner, so it ends up poisoning the machine that the normal protein would build if it could. Given that, we often refer to these as dominant negative alleles, interacting alleles, interference alleles. There are a lot of different descriptions for them in function space, but Muller's idea was that they are antimorphs. They poison a complex with a normal protein rather than allowing that complex to form and have its role. These are often dominant in the presence of a wild type allele because if the mutant protein assembles with the mutant protein, you have a broken machine. If the mutant protein assembles with the wild type protein, you have a broken machine. And only in the rare circumstances where two wild type monomers come together to form the machine do you get a tiny little bit of function. So oftentimes the antimorphic allele is dominant to the wild type allele. Neomorphic alleles are fascinating. These are mutations that change the molecular identity of the gene that they happen to. A famous example of this is the lens crystalline that functions in light focusing within the lens of our eyes. This, it turns out, is an ancient liver enzyme that picked up an expression pattern where it began to develop in developing eye tissue. At high concentrations, it forms a paracrystalline type of structure that is useful in focusing light. This is a mutation in which you changed where a gene is expressed. Rather than a liver cell, it's now expressed in cells that are contributing, epithelial cells that are contributing to the development of the lens. Because of this neomorphic mutation, expression in an ectopic place or time, a place in the body where it isn't normally expressed, this became a new gene. It transitioned from having its liver enzyme activity in the liver, playing a new role in the eye. The ability to focus light is a very powerful selective advantage for those organisms that have it, and thus this new gene kind of took off on a life on its own as a gene expressed in the eye. So we describe alleles like this as neomorphic. They're mutational changes in which now the gene is truly doing something brand new. So if we transition to the next slide, we have an interesting situation here where we're kind of talking about the molecular products of different alleles. A wild type allele, upon transcription and translation, makes a wild type protein. That red squiggly line is the wild type protein doing its normal job. If it should have been a screwdriver, it's a great screwdriver. It's screwing in the screws in all the cells. That gives us a wild type idea. A recessive amorphic mutation, complete loss of function in a cell that's homozygous for the amorphic mutation. That cell has no screwdrivers. If screwing in screws was important for that cell, necessary for its life, that cell is not going to make it. A hypomorphic mutation, it's a partial loss of function. Rather than a screwdriver, maybe we make a screwdriver that has a very blunt blade. Maybe that blunt blade can only fit in the slot of a few screws, not all of the screws it used to fit in. Some screws are getting screwed in, but not all the screws. So the cell has a ability to survive, but it's kind of beat up. Its activity isn't what you would expect because it's homozygous, shall we say, for a hypomorphic mutation. Neomorphic mutations doesn't look like the wild type protein at all. It's doing something brand new. Rather than being a liver enzyme, it's an eye protein enzyme. Rather than chewing up nasty organic chemicals in the liver, it's now focusing light in the lens of the eye, making sure that we have a focused image on our retina. The mutation, as it originally arose, it arose in a gene that had a history of a different biochemical activity, but now has an entirely new function in the cell. Oftentimes you have novel mutant phenotypes. These are often dominant in relation to wild type alleles, but it really depends on what the interactions are. And then in the next slide, I just kind of go through possible dominant interactions. Often the wild type allele is dominant to amorphs or hypomorphs, but in the case of neomorphs, the neomorphic allele may be dominant to the wild type protein. Okay, so we've really talked quite a bit about alleles. Now let's talk about interesting allele interactions, where we have situations where there is not pure dominance or recessiveness, but you have some range of interactions in terms of how these alleles influence each other and how their presence in the cell contributes to the phenotype. So the title for this slide and the start of this section, The Wild and Wacky World of Allele Interaction. So patterns of dominance between alleles. Here I'm just going to describe briefly some of the alternatives. In the case of complete dominance, which is mostly what we've discussed up to this point, one allele completely obscures the presence of another in the genotype. So if, for instance, you're big A, a homozygous dominant, or you're a heterozygote, big A, little a, your phenotype is driven by the dominant allele. We would describe the phenotype as big A, because although the recessive allele is present in the genotype, it doesn't have an opportunity to express itself through the phenotype because of the presence of, on the other homologue, of the dominant allele. Incomplete dominance is a fascinating situation. Both alleles have some impact on the phenotype. That means usually there's a distinct phenotype associated with every genotype. If you're big A, you will have one phenotype. If you're big A, you will have a different, distinct phenotype, often intermediate between the big A homozygous and the little a homozygote. Similarly, the little a homozygote will have another distinct phenotype. So when you have incomplete dominance, often the allele associated with one extreme, often what you see in the wild type, is seen to have an influence in the heterozygote, but so is the mutant allele. The mutant allele will also in the heterozygote have some effect. Thus we can say the phenotype of the heterozygote is a collaboration between these two alleles in a situation of incomplete dominance. They are influencing each other. They give a unique third phenotype that you don't see in either the little a or the big A individuals. Now, that's going to sound very similar to the phenomena of codominance, but in fact we can think about how they're distinct here in just a bit. In the case of codominance, you will have alleles that have different designations. Usually here they're using the allele designations A and Q. That can be confusing because it may sound like we're talking about two genes, but in fact we're talking about different forms of the same gene. The A allele of gene 1 or the Q allele of gene 1. So here, what are the possible genotypes we see? We see big A, big Q, heterozygous, big Q. Now in this case we also see three phenotypes, but we don't see an intermediate phenotype here that is truly distinct. Why? If big A gives us a big A associated phenotype, big Q gives us a big Q associated phenotype, the heterozygote shows both phenotypes fully. So now let's think about that. Big A might be a very long branch. Big Q might be a crooked branch. If you're big A, big Q, you're a very long crooked branch in a plant. We see just by looking at the phenotype, oh, that individual is a heterozygote because we see both the influence of A, the branch is long, and the influence of Q, the branch is crooked. So with codominance, we see phenotypes in the heterozygotes in which each allele is fully expressed. That's different in incomplete dominance, where we see that the phenotype is an intermediate collaboration. Both alleles are influencing the ultimate readout of the phenotype. We don't see them both distinctly. We see only that there is a collaboration between them. Now, how do we describe dominance interactions with codominance? It's a bit sticky. We describe in the heterozygote both alleles are dominant. Why? We fully see Q, we fully see A, both alleles are dominant in that situation. Okay, folks, so one of the things I'd like to do now as we kind of transition to thinking about different possibilities for how alleles interact is actually kind of talk a little bit more about this idea of complete dominance. What are the different possibilities for how dominant alleles exercise their activity in a heterozygote? How are they able to conjure up a wild-type phenotype given the presence of potentially one complete loss of function allele and one normal functioning allele? Well, one of the things we have to introduce here is the concept of a threshold for biological activity, or a threshold requirement for a gene's function. There are sort of two different ideas about this concept of a threshold. One is that you need enough of something, either enough of a gene's protein product and the activity of that protein within a cell, or potentially, let's say that that protein product is an enzyme, you need enough of the ultimate substrate to product conversion of that enzyme, i.e. the product has to accumulate within the cell to a level that is required for the cell's normal life and function. So with heterozygosity, we have two sort of interesting possibilities for how dominant alleles can be dominant. One possibility is that only one copy of a gene is sufficient to accumulate enough of that gene's encoded protein or to accumulate enough of the ultimate product of that gene's function. Let's say in substrate product conversion by an enzyme, enough of the product for the cell to have normal life and physiology. In a heterozygote, you may have less of the wild type function. Let's say each allele contributes 1/2 of the total cell's amount of the activity of gene A. Well, in a heterozygote, you would only have half. In a homozygous dominant, you would have 1, you would have a full dose. And if you are homozygous recessive, you would have no or very little. So the homozygous recessive situation, clearly the activity of that gene would drop below the threshold requirement. In a heterozygote with half, that might be sufficient if the gene, if that level of the gene's function is enough to get past the threshold requirement. Let's say the cell's threshold requirement for that gene's activity is 1/3 of a total amount of 1. Well, if one wild type allele gives you 1/2, that's greater than 1/3, and thus the cell is able to do normal life and activity. So in that case, a dominant allele is dominant because it provides enough of a dose of wild type gene function that the cell's needs are met, normal activity occurs, and because of that, the cell appears wild type. Now, if you actually measure the output of that gene, you'll notice that it's significantly reduced in a heterozygote in the first case, but still there's enough. The other possibility for how alleles can be dominant is that the allele undergoes some type of re-regulation, that in a situation where you have 2 wild-type copies of the gene, you are homozygous dominant, you produce an amount equivalent to 1 dose, half from each gene. If, however, in a heterozygote you have at least 1 wild-type allele, it may be normal, it may have as a partner a complete loss of function allele in the heterozygote, but that one allele may now be re-regulated so that it in effect fully compensates for the loss of 1 dose of the gene. Thus, if you were to measure the amount of enzyme activity in the heterozygote, it would be the same as in the homozygous wild type. If you were to measure the amount of protein product generated through transcription and translation by the gene, it would be equivalent in the heterozygote or the homozygote. So this is only important because later on we're going to think a little bit more quantitatively about gene activity, how much gene activity is required for wild type. And this just shows there are 2 alternative ways to be dominant. In one situation, a single wild type allele produces enough, although less, enough material or activity for the cell to do normal physiology, normal function, normal activity. And so even though heterozygotes have less from an objective standpoint, they have enough. That's in contrast to a situation where there's re-regulation, in which the wild type allele is in some ways amped up. It's caused to now produce the equivalent amount of gene activity, molecules, functions, etc., however you want to define that, as a cell would if there were 2 wild type copies. Either of these situations can result in dominance. The wild type allele controls the phenotype, but they do it through different biochemical schema, different biochemical mechanisms. All right, so now let's explore the different possible allele interactions, and let's start with incomplete dominance. So here we'll begin with the slide, the paint bucket problem. Mother Nature throws Mendel a curve, but he hits it out of the park, incomplete dominance. So this is a famous example with the color of snapdragon flowers. So there are true breeding lines, again like peas, there is a snapdragon fancy, there are collections of hobbyists that work together to preserve different types of genetic material to grow different plants. And you see polymorphism, you see distinct phenotypes in some of these different true breeding homozygous lines. A fascinating cross was done in which two different true breeding lines, a true breeding line that only produces white flowers and a true breeding line that produces deep red / crimson flowers, were crossed together. This experiment was done with the expectation that we would see simple dominance, and if in keeping with the situation in peas, we might assume that red would be dominant to white. Under that kind of hypothesis, crossing homozygous dominant red by homozygous recessive white, we would expect our F1 heterozygotes to be red, and then if we crossed those heterozygotes together in a monohybrid cross, we would anticipate a 3-to-1 phenotypic ratio, 3/4 red to 1/4 white, in the next generation of plants if we just continued doing that cross. What was surprising is this was not the result that was seen. When you cross these two true breeding lines together, you see instead that the F1 produces a unique phenotype not seen in either parents. The plants now have pink flowers, intermediate in intensity between red and white. Thus, we see an instance here where the phenotype begins to favor one parental allele. There is color in contrast to no color white flowers, but it simply is the case that a single wild type allele is not sufficient to bring the pigmentation level past the threshold for red color as detected by our eyes. So we describe this as a phenomena of incomplete dominance. Homozygous dominant, big A, if you will, or big R, flowers are red. Homozygous recessive, little r, flowers are white, and heterozygotes are intermediate between them. The interaction is one of incomplete dominance because the red favoring allele does not fully obscure the presence of the white favoring allele in the heterozygote. Now, if you take those F1 heterozygotes and do monohybrid crosses, big R by big R, in the next generation we see Mendelian ratios coming back. So we were a little concerned about what we might see. Is Mendel still there? Is this a situation where we're just going to blend away? We're never going to get our parental red and white back? No, we see red flowers reemerge in the next generation. We see pink flowers again reemerge, and they in fact are the most frequent progeny class. And then we also see white flowers reemerge. So the alleles are still there. They're still durable. They still must be segregating in terms of meiotic segregation. There must be statistical independence in patterns of gamete fusion. All of our expectations from a monohybrid cross are here. But what is surprising is that the intermediate phenotype is the phenotype that is most common. Why is that? Well, it goes back to our expectations about the Punnett square that we discussed when we talked about tall and dwarf alleles for plant height. There are two alternative, mutually exclusive patterns of gamete fusion that give us heterozygous offspring in a heterozygote by heterozygous cross. One of those, the red favoring allele, came in through the pollen. The other, mutually exclusive heterozygous genotype, the red favoring allele, came in through the ovule, through the female gamete. And thus, because there are more patterns of gamete fusion that build heterozygotes than homozygous red or homozygous white. It is not surprising that the heterozygote is the most common genotypic and phenotypic class. So given that type of expectation, we now have an interesting situation. We can go, okay, well, from what we already know about heterozygous by heterozygous crosses, we would expect 1/4 of the progeny offspring to be homozygous dominant, 1/4 homozygous recessive white favoring, and 2/4 or 50% to be heterozygotes. Does the actual cross result meet that classic model? How would we describe that phenotypic expectation? Rather than 3-to-1 dominant to recessive, we would expect 1 to 2 to 1. 1/4 homozygous red, 1/4 homozygous white, and 2/4 heterozygotes. That would be our classic statistical model for describing this phenomenon. We can now ask the question, if we do a frequentist study, we do a real experiment, is that what we see? Do we see in our real data that conforms well, is a good match to our classic model? Well, not surprisingly, these types of experiments have been done and done quite frequently, and if you look at the resulting data, we see some reassuring events. If we kind of move into the next slide here, where we have red, big W, by white, little w, heterozygotes in the F1 that are self-fertilized, we see a phenotypic ratio in the F2 progeny that's interesting. We see 62 out of the total that are red, 57 out of the total that are white, and 131 out of the total that are pink. Here we have our frequentist data. Is this a good match to a 1 to 2 to 1 expectation? Well, in order to test the goodness of fit of our data, we can do a chi-square analysis. In this case, how will we build our theoretical classic data set? Well, we know how many flowers in total were counted, or how many plants in total were counted, 250. Under our model of 1 to 2 to 1, we would expect, rather than 62 red plants, we would expect 62.5 red plants. Rather than 57 white plants, we would expect 62.5 white plants. Now, remember, in general, if it's 5 or above, you move to the next higher whole number. If it's 5 or below, you move to the next lower number. Of course, here we have precisely 5. I'm just going to assume that we can score a half plant in this case, but in general and on my test, remember, if you're 5 and above, move to the next whole number in terms of the number in a phenotypic class. If you're below 5, move to the next lower number. So now here we have an idealized data set fitting a 1 to 2 to 1 ratio. 1/4 of 250, 62.5 reds are expected. 1/4 or 62.5 whites are expected, and 125 pinks are expected. We can now ask the question, does our real world data set, 62 to 131 to 57, closely enough match our classic data set, 62.5 to 125 to 62.5, such that we don't have to reject the null hypothesis that these two data sets are essentially the same, and that our null hypothesis, this is 1 to 2 to 1 segregation, these are not different, does not have to be rejected. Well, we can now run these new observed numbers and expected numbers through a chi-square analysis shown below. 62 minus 62.5 squared divided by 62.5. 131 minus 125 squared divided by 125, and 57 minus 62.5 squared divided by 62.5, and we can get our summed chi-squared value. Here, how many degrees of freedom would we have? Well, we see 3 potential phenotypic classes. By convention, our degrees of freedom are number of phenotypic classes minus 1, so 2. I'll let you guys track this down in your book's distribution of p-values for the chi-square analysis, but with degrees of freedom 2 and a summed chi-square value of 0.776, I think we can be confident that we're going to have a pretty high p-value. It certainly won't be less than p equals 0.05. Thus, my estimation is, just by eyeballing it, we're going to find pretty good goodness of fit between our classic model expectations, 1 to 2 to 1 segregation, and our real-world observed data, our frequentist data set, 62 to 131 to 57. Given good concordance, this is reassuring to us that what we're seeing is a phenomena like incomplete dominance. We're seeing heterozygotes at the frequency that we expect and homozygotes at the frequency we expect, but in this case, the number of dominant red-favoring alleles matters. If you are homozygous for those alleles, you are red. If you are heterozygous, you are pink. Thus, the phenotype really is a collaboration at some level of these two values. The intermediate favors one of the parents, the red favoring parent, and thus we describe this as incomplete dominance, not codominance. It would be codominance if we saw fully white and fully red in the F1. But because the F1 is pink, we say this is incomplete dominance, not codominance. We can tell that they're heterozygotes, but we cannot see fully in that phenotype the white allele controlling part of the phenotype and the red allele controlling part of the phenotype. That phenotype is different than a long twisted branch in which we could see both branch shape alleles fully expressed. So this is different than the situation we have with codominance. With codominance, we really can say, if we move to the next slide here, titled Codominance, open quote, both alleles are dominant, closed quote. Here we're going to see a different phenomena in terms of allelic interactions. Here, when each allele is present in the genotype, both alleles will be fully seen, fully expressed in the phenotype. Thus, we can shorthand this as both alleles are dominant. This highlights the fact that for single genes, there can be many alleles that exist in populations. Individuals, usually being diploids, have 2 of the alleles, but populations can harbor many alleles. You may have a huge number of possible homozygotes and heterozygotes, given the many different ways that DNA can be altered and the many different ways that those DNA alterations can feed into RNA alterations and ultimately into protein alterations. So populations can harbor quite a number of alleles, and we will often see a very diverse, extensive array of genotypes and phenotypes. The human blood antigen system is really a beautiful example of a system that has a number of different allele interactions in it. Codominance is a huge part of it, but we'll also see that there is room within this blood system for simple dominance and recessiveness as well. Some of the alleles will show codominance. Other types of alleles will simply be recessive to other alleles. So we're going to kind of see a whole array of possibilities for allele interactions. The gene under consideration here is the isoagglutinogen locus, or the I gene. The isoagglutinogen locus is responsible for synthesizing a sugar moiety attached to proteins on the cell surface of many cells, but also red blood cells, and that by changing different sugar types on the surface, they change the immunogenicity of that cell. How your own immune system recognizes or does not recognize a cell as self, as part of your body versus a foreign invader. So the I gene plays a very important role in molecularly decorating, adding different sugar types to the surface of cell-associated proteins. So surveys of human populations that we've seen have revealed 3 alternative forms or alleles for the isoagglutinogen gene. You have the I superscript A allele, also called by shorthand A. You have the I superscript B allele, also called by shorthand the B allele. And you have the I superscript O allele, also by shorthand called the O allele. Each of which encodes a different isoform, a different morph of the isoagglutinogen gene, and those different morphs result in different types of decorations in terms of sugars on the surface of cellular proteins and other molecules. So this is basically a sugar adding enzyme. The A form adds one type of sugar. The B form, an alternative type of sugar. The O allele cannot add sugars to any surface antigens on cells. So with multiple alleles, we have sort of an interesting situation. As the number of alleles increases, the number of possible genotypes begin to increase. Let's just kind of briefly run through the 3 alleles that we've discussed so far. With A, what are the possibilities? You could be AA, homozygous, an AB, heterozygote, an AO, heterozygote. Those are the possibilities for A. With B, what are the possibilities? You could be BB, BO, BA. With O, what are the possibilities? OO, AO, or BO. So here we're in an interesting situation with 3 alleles. We know we can generate a maximum of 6 potential genotypes. Now, any individual can only have 2 alleles. A person may be AA, a person may be AB. A person can't be ABO because they wouldn't be diploid, they'd be triploid. They would be a new variant of the human species. And so we're in an interesting situation where we have multiple alleles, but any one individual being diploid can only have a genotype composed of 2 of them. So there, the frequencies of alleles in populations drives in part the genotypes that you will actually encounter, but given 3 alleles, it is possible to construct 6 genotypes. I've given you a little shorthand equation here in the table at the bottom of the page. N times n plus 1 divided by 2. This should be the number of genotypes. So starting with n alleles, let's say 6. How would you calculate the number of genotypes? You would calculate it 6 times 6 plus 1 or 7 divided by 2. With 6 alleles, you can easily use this equation to calculate the number of possible genotypes that can be made, even though in individual populations it depends on the alleles are there what is actually possible. So if we now kind of run through the blood type system, we'll do this fairly briefly here before following up, and I may also begin with this next Tuesday. The IA allele, if we move to the next section, section 4.5, the next slide, the I superscript A allele is responsible for an enzyme that can add a terminal sugar N-acetylgalactosamine, also called GALN, to the H substance, which is a core sugar molecule, which can be attached to other cell surface moieties, proteins, other molecules as well. The B allele produces a variant of the sugar addition enzyme that adds a terminal galactose. So if you're the B form of the enzyme, you add galactose to this core sugar substance called H. If you're the A allele, you add N-acetylgalactosamine to this core sugar framework substance H. So H either gets decorated with N-acetylgalactosamine or gets decorated with galactose. The O allele is interesting. It is a defective form of the enzyme that cannot add either terminal sugar to H. So if you are OO, your surface cell surface sugars are just the H substance. They don't have galactose on them or N-acetylgalactosamine. Only H is found on the surface. Now, why does this matter? It turns out that these surface antigens are very similar to sugar antigens that are on a variety of different soil bacteria. Many of these soil bacteria inoculate babies very early in their life, often as they leave the birth canal. Thus, you'll have bacterial inoculations in which babies will be exposed in their bloodstream to A surface antigen or B surface antigen, A and B, on these bacteria. Now, a baby that has AA blood can develop antibodies as part of the adaptive immune system, part of the acellular immune system, to anti-B targets. So they can develop anti-B antibodies to help clear their systems of infections with B-bearing, with bacteria that bear the B surface antigen. However, AA babies can't develop anti-A antibodies or they would destroy their own red blood cells and body tissues. So this is interesting. AA individuals very quickly develop anti-B antibodies. Similarly, individuals that are BB develop anti-A antibodies but cannot develop anti-B antibodies, or they would kill themselves. They would end up having a crisis that resulted in their death. You can't destroy your own organs. That's a no-no. Individuals that are O are fascinating. They can develop antibodies against bacteria waving the B sugar around. They can also develop antibodies against bacteria waving the A sugar around. And so individuals that are O can definitely develop an immune reaction to any bacteria that is displaying either of those antigens on its cell surface. Individuals that are AB are interesting. How do they clear bacterial infections? They can't develop antibodies against A, they can't develop them against B, but luckily these soil bacteria have many other surface antigens. So individuals that are AB heterozygotes will develop antibodies to help fight off bacterial infection, just not anti-A or anti-B. So now let's go through the ABO antigen system. Here we can kind of see the one dive into organic chemistry we're going to do this semester. We can see a little bit about what's going on with ABO. I think I've gone a bit long at this point, however, so why don't we pick up from this slide, the ABO antigen slide, next Tuesday. Thank you, my friends. Have a good day. Bye-bye.